Hi everybody, and welcome to Cell-fie Science, where I bring you a quick dose of science from my cell phone. I'm Jen, a PhD candidate studying plant biology, and my particular research interest is in cold stress. So that's where the humming's coming from behind me. I'm running a freezing assay inside this glycerol water bath. Now the glycerol acts as antifreeze to keep the water from freezing because this is actually running at -3 degrees Celsius. Earlier this morning I used this core to make leaf discs out of my plants. So I took the leaves off and I just used this like a hole punch so that I could generate a whole bunch of leaf discs. These leaf discs were then put into test tubes, which were put into the water bath starting at -1 degree Celsius. Right now they're at -3, and let's see if you can take a little peek in here. So that's what they look like. You can see the leaf discs inside the tubes. Now I'm running this until -4 degrees Celsius, and I'm lowering the temperature every 2 hours by half a degree. This assay is going to take me 14 hours to complete. I'm actually going to be here until 12:30 a.m. and I started at 9 a.m. So it's a very, very long assay. So why do it? Hang on, I'll go somewhere a little quieter and I'll explain. And I'm back, broadcasting from one of our teaching labs now. So we're talking about freezing, and one of the things you might have noticed about water is that when it freezes, ice floats, right? It's always found at the top of your drink, not the bottom. This is really unique because most things when they freeze contract, but water actually expands. It gets bigger and it gets lighter and it floats to the top of itself. It is less dense. Cells are made mostly of water. So now let's take a look at my very poorly drawn plant cell here. And you can see that we have the plasma membrane in black. Now that's the sac, that's the membrane that surrounds the cell. Kind of like the outside of a water balloon, it keeps the water inside the balloon separate from maybe the water around the balloon as you're filling them up and throwing them in a bucket. So that's the plasma membrane, keeps everything inside. Plant cells also have the unique cell wall, right? That's in red. And the cell wall is kind of like armor, just keeps things a little more rigid, a little more structured, and make sure less damage. Filling our plasma membrane is the cytosol, and cytosol is made mostly of water, but it also has things like electrolytes, proteins, signaling molecules, the stuff the plant might need to get to A to B. Now you'll notice the plasma membrane and the cell wall in my drawing are not touching, and that space between right here is called the apoplast. Alright, that's an important piece of anatomy for what I'm doing next door, because when freezing happens in a plant, it typically starts in the apoplast. So that's why I have highlighted this section. Now, because they're mostly water, what's going to happen as the temperatures drop is ice is going to start to form. Alright, so here's our little piece of ice right here in the corner. As the ice forms, it can grow, and its sharp little edges can poke our plasma membrane. Or maybe they even burst through the cell wall. As that happens, all that cytosol is going to be leaving the cell. So what I'm going to do tomorrow is I'm going to take my leaf discs and put them in some water that's completely devoid of any kind of electrolytes or proteins or any of that stuff. And then I'll put my leaf discs in that water and shake them for a really long time. After the shaking, I'll read them on a special conductivity meter, which will let me know how much electrolytes were in the solution. So a plant that is very good at adapting to the cold will have less electrolytes because less ice will have formed, so less damage will have formed. Where the ones that are more susceptible to cold will have more ice, which will lead to more cold damage, more holes, more electrolytes, higher and higher it will go, and I can measure that. So what I will do is I will take all my data from tomorrow and I'll put it in a graph that will probably look pretty similar to this one. And you can see down here we have plants with very low electrolyte leakage. Alright, so these are very, very little leakage coming out. These would be cold tolerant plants. Where these ones up here have a lot more electrolyte leakage. This is actually 100% electrolyte leakage, so all of the cells have burst. There's nothing left except cytosol floating around randomly. So these would be very cold susceptible plants. And that's how I can use freezing to measure cold tolerance for our plants. So that's it for this episode of Cell-fie Science. I'll see you next time.